last talk, I realize I'm in the uncomfortable position of standing between you and a beer. So I'm going to get right to it here without any jokes. And my topic tonight's going to be about teamwork in scientific discovery at the university. And there'll be some science, but I'll also want to talk about how the students are integrated into this. So much so that when I won the prize and was able to take some people along to Stockholm, I took one of my graduate students, Paula, there on the left, her husband's on the far right, my long-term research associate, Art, who's on the second from the left, and his wife, who's on the second from the right. So this is how much this teamwork means to us. Now, the particular scientific topic tonight is going to have to do with the ends of our chromosomes, which are called telomeres, and these are seen here in this micrograph of human chromosomes, shown in blue, as these little yellow dots. So we're lighting up that there is something common at the ends of all of our chromosomes. And it turns out that this is a particularly vulnerable part of the chromosome, and it needs to be protected, sort of like the little plastic tips on the ends of your shoelaces. Protect your shoelaces from unraveling. So think of the shoelace as the DNA and the little plastic tip as this telomere. Turns out that in adult cells that have quit growing, the ability to build out the end of the DNA is turned off, and the chromosome ends actually shrink with successive divisions of the cell. And when they get short, then that signals the cell to stop dividing. And that's a good thing, because once you're a full-grown person, you don't want all your cells to keep dividing, right? You'd get really big really fast. So that's a protective mechanism also against cancer, because one of the features of cancer is that it's uncontrolled cell proliferation. And in fact, tumors stumble on tricks to reactivate the process that extends this telomeric DNA so that they can be immortal, much to our detriment. Now, what is this molecular machine that turns on that replicates or builds out the ends of the chromosomes? It's called telomerase, and it was discovered by Liz Blackburn and Carol Greider, who got the Nobel Prize a few years ago for this discovery. And they also found that, quite remarkably, and different from a lot of enzymes in cells, it has an essential RNA, or ribonucleic acid component, shown here as the purple ribbon. And one of the functions of that RNA is that it acts as a template for telomeric DNA synthesis. You all know your DNA or RNA alphabet. A pairs with T, C pairs with G. Well, here the A's and C's on the telomerase RNA specify the T's and G's on the end of the chromosome. And the way that this works is that the RNA template is used as sort of a mold. The DNA is built out on that, and when it gets 6 bases down, it releases, ratchets back, and starts all over again, and you get this successive extension of chromosome ends. Now, this RNA part of telomerase, though, isn't the whole game. There's another essential component. It's a protein shown here as a yellow egg, and despite intense and very competitive action around the world, for 10 years after Blackburn and Greider discovered the RNA component, no one could find the essential protein component, which, by the way, was going to end up being the key to cancer, because the RNA part is present always in our cells, but the secret for cancer activation lies with the activation of this protein component. So at this time, a young Swiss PhD student who had just gotten his PhD in Basel, Switzerland, came to my laboratory, Colorado, and decided to take on the daunting task of discovering this protein component. And we talked about which organism we should use, and we decided on this fellow, which you're all very familiar with, right? This is Euplotes ediculatus. You've all heard of this. Also known as part of pond scum. You can see some of its friends from the pond scum talking to it there. This can be found in Varsity Pond at CU Boulder. Not kidding. And why would we choose, this is a cancer project. Everybody else was looking in tumors for this enzyme. Why would we look in this guy? Because each of you has 23 different chromosomes. Remember 23andMe. And then you've got 2 copies of each of those, 46, 2 ends for chromosomes. That's about 100, right? This guy has 100 million tiny chromosomes per cell. So there's about 1 million-fold more chromosome ends than any of you have in a cell, and we thought that this would give us a head start on this project. So Lingner was actually able to do this, and then was able to leap, after spending 3 years finding the Euplotes telomerase, leap to the human version of this in a few minutes by using the Human Genome Project, because in evolution, often things are invented just once in biology, and if it's a good solution to a biological problem, it's repurposed, or as we would say in Boulder, recycled endlessly through other organisms, and so the human gene was then found in the computer. Now, let's get back to cancer cells for a minute. How then do they reactivate this telomerase protein? And this was figured out by my friend Levi Garraway at Harvard, who discovered that in many, many different cancers, just a little bit ahead of the gene, in the gene regulatory region, the cancer cells had lucked into making a mutation which then activated the synthesis of the messenger RNA for that gene. And this is a remarkable mutation, the third most frequent in all of cancer. In fact, 80% of all the melanoma tumors from diverse places around the world have independently acquired this same mutation. 80% of this deadly brain cancer, glioblastoma, 70% of bladder cancer, etc. And other cancers, very interestingly, don't have this mutation. We're still working to figure out how do they activate. They still activate telomerase. They do it in some way other than this regulatory mutation. Now, this really hit home for me personally when a dear friend of mine emailed me and said that they had had their tumor DNA sequenced, and that the gene that we had discovered and the mutation that Levi Garraway had discovered in the control region was one of the top things that differentiated their tumor from their normal cells. And this was a sad moment because the people who had sequenced, the company that had sequenced the tumor DNA, very properly and correctly said this TERT promoter mutation is thought to be a driver for cancer, but there are no approved therapies for this, and scientists are working on it. So we are, in fact, working on this, and we're using a team approach. And we not only work together in the laboratory, but we hike together in the Colorado Rockies in the summer. And I want to particularly point out that a number of the people in here on the lab hike and in our team at CU Boulder are undergraduates, and particularly Caitlin Bell in the green shirt, who came from Denver, started working in my lab as a freshman and continued through all of her years, went on to medical school at Vanderbilt, then research at Harvard, and now she's at Stanford. And so she was so successful in the laboratory that she was, in fact, the second author on this breakthrough that we made, where we found a patch of a protein that's on the end of the chromosome, the telomere, that attracts or recruits the telomerase enzyme. And now we're working to find ways, if we could interrupt that attractive feature, that would be a way of preventing the telomerase from working in tumor cells. So my final point, another shot of one of our laboratory hikes, is that people often ask me, well, how should we balance teaching at the university with the time we spent on creative activities like research? And I think that's the wrong question, because if you actually live this life that I live, the beauty of it is that those are not two separate things. The education and the research are not things that we can wall off from each other, but they're really integrated. And it's the students in the laboratory who are helping us do the research, and it is doing the research that really engages the students and makes their education so valuable to them. Thank you very much.